Hello, I'm Dr. BCMR, Assistant Professor at Shastra. Genome, which is comprised of DNA, takes several shapes and numbers when we account for the differences in the genetic information of diverse organisms, starting from, say, acellular propagating entities like viruses or plasmids, to that of complex or multicellular eukaryotes like us. And that is what we will try to appreciate in today's talk. So this shape itself, it could be circular or linear. Say in majority of the prokaryotes, the genome is circular, whereas in the eukaryotes, its majority of it is in linear. And then in majority of the prokaryotic genomes are single. Say for example, E. coli, the gut bacterium from humans or many of the mammals, they have a single circular chromosome. Whereas you can also take another bacteria, for example, Vibrio cholera, that causes cholera, has two circular chromosomes. So in prokaryotes, they may have more than one chromosome, but majority of them are circular. If we take eukaryotic chromosomes, majority of them or it's general that they all have numerous linear chromosomes. So these are some of the differences between prokaryotes and eukaryotes. They have similarities and they have differences as well. Next, we also have some other entities such as plasmids, mitochondrial DNA and chloroplast DNA. Say, if we go by the traditional definitions, we call plasmid as a dispensable extra chromosomal material, and says it is almost like that of mitochondrial DNA. Only the difference is not all bacteria have plasmids. Some of them have and some of them don't have. In my perspective, I consider plasmids as molecular parasites very much similar to that of viruses. Viruses infect a cell, they will propagate and they will move on to other cells. It is almost the same with the case of plasmids. They infect a cell, they use the cells for their own propagation. So the only difference, if there is any, between viruses and plasmids is that viruses have more than just DNA. They have proteins when they are formed as virions. Plasmids, as far as we know, they exist just as the nucleic acid, which is the physical form of the propagating entity and also the genetic information. So I don't see any difference in that way. When it comes to mitochondrial DNA, all eukaryotes, there are exceptions, but as a general rule, all eukaryotes have mitochondrial DNA. And why do I consider it as a part of the genome or not? Are the same is the case with chloroplast DNA. Chloroplasts also have circular DNA. Chloroplasts and mitochondria have apparently, or as far as we know, that we can believe that they have arisen from bacterial origins, or they have bacterial origins. So if I take a plasmid out of the cell, out of a bacterium, the bacterium survives and the plasmid can survive in the sense it still retains the potential to infect some other bacterium host and then propagate again. If I do the same thing with mitochondria, I take out the mitochondria from a eukaryotic cell, the eukaryotic cell is going to die because it is not able to efficiently produce energy. The mitochondria cannot survive because majority of the proteins that are synthesized, that are mitochondrial, they are encoded by the nuclear genes. So in that way, in that sense, using this kind of logic, we can say that mitochondria is an integral part of the eukaryotic cell. We cannot or don't have to say the same about the plasmids. Plasmids can be removed and then they still can exist and manifest their propagation when opportunity arises. And bacterium can survive if the conditions are good enough. So plasmids are separate entities. very much similar to that of other parasites like viruses. So if we move ahead a little more, we will try to understand the different sizes and shapes. We will see a few exciting results or observations here. E. coli has one chromosome. It is about 4.6 MB. MB is for megabase pairs. MBP, you can say, MB for short. And 1000 base pairs is equal to 1 KB, and 1 KB is equal to 1 MB megabase pair, and 1 MB is equal to 1 gigabase pair. So 1 gigabase pair is about 10 to the power 9 base pairs. That is what we have in our genome as well. We have about 3.2 gigabase pairs. Okay, budding yeast has 16 chromosomes and about 12.1 MB, and fission yeast has only 3 chromosomes and 12.5 MB. You can notice that the chromosome number is different, but then the size is almost the same. Either we don't know which one is previous ancestral to which one, or the lineage. It seems like there has been some either fusions or breaking of chromosomes, but we can appreciate that the total genome is almost the same. Arabidopsis thaliana, it has 5 chromosomes and 125 MB. It's a plant. And fruit fly Drosophila melanogaster has 4 chromosomes with 180 MB. You can see about humans. We have in a haploid set, we have 22 chromosomes plus one of the sex chromosome, either X or Y. We are just talking about haploid, which we will discuss shortly. We have about 3200 MB, or we can say it as 3.2 gigabase pairs. Looking at this table, we could be convinced that we have the largest genomes, but that is not true. There are several other plants and animals that have gigantic genomes, and say for example lungfish has about 42 gigabase pairs, at least 10 times more than what we have. And lungfish also has some special characteristics or feats that it can do. It can live in the soil embedded, dormant, when there is drought, away from the water. When there is no longer water, it can stay up to 4 to 5 years in a dormant stage. It's amazing how they can make that happen. Okay, moving on, we will try to look at the chromosome numbers and how it relates to us in cells. We are all diploid. Diploid is about having 2 sets. Haploid is having 1 set. We'll say it as N, whereas diploid we will say it as 2N. So here there is a green chromosome. There are 2 green chromosomes and 2 blue chromosomes. So in this, the N would have consisted of 1 blue and 1 green chromosome, and we have 2 of each, so we have 2N, that is diploid. All the body cells that we have apart from the gametes are diploid. There are few exceptions we will see shortly. In haploid, we only have 1 set, that is 1 green chromosome and 1 blue chromosome, and that we call it as haploid. The gametes, the mature gametes in us, which comprises the germ plasm, is haploid. It has only 1 set of the whole genome that we have. There are some abnormalities that do occur in certain cases. You can see it. There are 2 situations called as polyploidy and aneuploidy. In polyploidy, we have 2N plus N, or that means we have an extra set of the whole genome set. So looking here, we have 3 blue chromosomes and 3 green chromosomes, which is making it triploid. We have 3 sets of N, that is triploid. There are also tetraploid as well, or you can have multiple ploidies like this. This is much more common in plants. So the endosperm is triploid. and that's a normal part of the tissue when it is associated with reproduction. And tetraploidy is also common in several plants, in the sense that the new species do arise because of polyploidy. And in humans, if it is, it would have been an abnormality, in animals at least. Or most cases it's abnormality, but in plants sometimes they do exist. There is another situation called aneuploidy, which is having less or more of one or more chromosomes. We are not talking about the whole sets, but some missing some chromosomes or having additional copies of some chromosomes. This does happen because of cell division issues that will be discussed at a later time. But right now, if we can appreciate what is the difference between polyploidy and aneuploidy, that will be sufficient. In polyploidy, it is about having a whole set extra or more sets, whereas in aneuploidy, it's about plus or minus. Although indicated plus here, it could be missing some chromosomes as well. It's about individual chromosomes. So this is, we also have quickly, we will try to understand or try to recollect some of the things and review. We have discussed about nucleoid, or which is the bacterial chromosome along with the DNA, with other proteins is called as nucleoid, which is kind of analogous to nucleus in eukaryotes. Prokaryotes do not have nucleus. So that's a region along with the genetic material is referred to as nucleoid. Diploid and haploid. Diploid 2 sets, haploid 1 set. Diploid in majority of our somatoplasm, the whole body is made up of it, and the haploid is about the gametes, mature gametes. And here there is another term that is about homologues. Say this green and this green are homologous, in the sense that homologues, which sense that they have exact same genome set. They do have same order of genes, and the genetic content is also same, so you call them as homologues. And we have discussed about the polyploidy and aneuploidy, and then gene dosage is something else. So because we have diploidy, we are likely to have 2 sets of every gene, but because of sometimes it is important to learn about it, especially when it comes to epigenetics and X chromosome inactivation and such scenarios. Say for example, with regard to polyploidy in eukaryotes especially, it's important, or it's interesting to discuss a topic that is about the megakaryocytes that produce platelets. On a daily basis, many lakhs of platelets are synthesized into our blood, and their lifespan is short, so they will be degraded. Their platelets are very small, lipid bilayer membrane bound, and they do not have any DNA in them. They function and they die, they get degenerated, and then megakaryocytes keep producing a large number of these platelets. And so to keep up with the demand for production, they have 128 copies of each chromosome. Although not all of the genes present on these chromosomes are expressed, majority of the genes are for the survival of the cell. The expressed genes are for the survival of the cell and production of large number of platelets. And that is how polyploidy is also present in our body, and it does have certain roles, but there are large number of abnormalities that can occur because of polyploidy in general. I hope we have got a fair enough idea about the numbers and shapes of chromosomes within the genetic material within different types of life forms or cellular forms. Thank you very much. See you later.